Have you ever stopped to think about what actually makes you, well, you? Today, we're cracking open the instruction manual for every living thing on the planet. We're going to take a look at the incredible molecular blueprint that builds and runs everything from the simplest bacteria to us. And right away, that leads us to a pretty mind-bending puzzle. If you could take the DNA from just one of your cells, a cell so small you can't even see it, and stretch it out, it would be about 6 feet long. So how in the world does all of that information get crammed into such a tiny space? We're going to find out. Okay, let's dive right in. The best way to think about how life works is to imagine a massive construction project. And in this project, you've got two main characters, the master blueprint that has all the plans and the crew of builders who actually make it all happen. What we're talking about here is a field called molecular biology. It's all about getting down to the nitty-gritty, studying how these tiny molecules work together to store information and carry out all the jobs that keep a cell alive. It's literally the science of how life works. So here they are, our two key players. On one side, you've got nucleic acids. You know them as DNA and RNA. These are the blueprints. They hold all the plans, every single instruction. And on the other side, you've got the proteins. These are the workers. They're the ones hauling materials, putting up walls, and running the power. They do all the actual work based on those plans. And here's the really wild part. This unbelievably complex code is built from a super simple little unit called a nucleotide. It only has 3 parts, a phosphate, a sugar, and a base. That's it. And there are only 4 different bases, A, T, C, and G. Think about that. The entire encyclopedia of life is written with an alphabet of just 4 letters. Okay, so we've got this blueprint, this enormous library of information. But that gets us right back to our big question. How does the cell store all of this? How do you fit 6 feet of DNA into a microscopic nucleus? Well, first, the code is organized into that iconic shape everybody knows, the double helix. You have 2 long strands that twist around each other. But the really clever part is how they're held together. The letters, or bases, have a strict rule. A always pairs up with T, and G always pairs with C. This isn't just a random detail. This rule is the secret sauce that allows DNA to be copied perfectly. And here it is, the answer to our puzzle. It's a masterclass in packaging. The long DNA strand is wrapped around these little protein spools called histones. Then that string of beads gets coiled up, and that coil gets coiled up again and again, packed tighter and tighter until it forms this super compact structure we call a chromosome. It's an absolutely brilliant way to compress data. Of course, for life to continue, for you to grow or heal, this blueprint can't just sit there. It has to be copied, and copied perfectly, every time a cell divides. The cell has an amazing molecular copy machine to do just that. The way it works is just beautiful. It's called semi-conservative, which sounds complicated, but the idea is simple. When DNA copies itself, the double helix unzips, and each of the original strands is used as a template to build a new one. So every new DNA molecule is half old, half new. This is a fantastic way to make sure you get a near-perfect copy every single time. It's a real team effort, run by a crew of specialized proteins called enzymes. First, an enzyme called helicase comes in and unwinds and unzips the DNA. Then the star player, DNA polymerase, gets to work building the new strands. And finally, another enzyme called ligase comes in to act like molecular glue, stitching everything together. And here you can see that whole operation in action. This is called the replication fork. There's helicase at the front, plowing ahead and unzipping, and right behind it you have DNA polymerase racing along both strands, building the new copies. It is a stunning, high-speed dance happening inside your cells constantly. So we've stored the code, and we've copied the code. But how does the cell actually use it? How does it read one specific instruction from that massive library to build a protein? For that, it needs a messenger. This is the big picture, a concept so fundamental it's called the central dogma of biology. The information has to flow from the DNA blueprint out to the protein workers. The DNA master plan stays safe inside the nucleus. When a job needs doing, a temporary copy of just one gene, the specific instruction needed, is made. That copy is called messenger RNA or mRNA, and it travels out to a protein-building factory called a ribosome. Let's zoom in on that first step. It's called transcription. An enzyme called RNA polymerase zips along the DNA, finds the right gene, and makes a single-stranded RNA copy.
It's basically like going to the library, finding a giant ancient book, and making a quick photocopy of just the one page you need.
Now, the messenger RNA isn't working alone. It's part of a 3-RNA team. You've got the mRNA, which carries the message. Then you have transfer RNA, or tRNA. Think of it like a delivery truck that brings the right building materials. And finally, ribosomal RNA, or rRNA, which is a key part of the ribosome factory itself.
All right, the message has been sent, the delivery trucks are ready, and we're on the factory floor. It's time to translate the code from the language of the blueprint into the language of the workers and actually build something.
This process is called translation, and that's exactly what it is. The ribosome clamps onto the RNA and reads the code in 3-letter words called codons. Each codon is a command for one specific amino acid, which are the building blocks of proteins. The ribosome just chugs along the message, reading a codon, grabbing the right amino acid, and linking them all together into a chain. But here is the real magic. A simple chain of amino acids doesn't do anything. The crucial final step is that this chain has to fold into a very specific, very complex 3-dimensional shape. That final structure is everything. It's what determines what the protein's job is, whether it's breaking something down, building something up, or sending a signal. The shape is the function.
Now, this whole system is unbelievably precise, but it's not totally perfect. The blueprint is alive, in a way. It can get errors, it has repair crews, and understanding all of this has a massive impact on our world. Sometimes mistakes happen. A typo slips into the code, a letter gets swapped or deleted, or an extra one gets added. We call these changes mutations. And while some mutations can cause problems, they're also the raw material for evolution. They're the source of all the incredible diversity of life we see. But your cells are on the case. They have an amazing toolkit of repair enzymes that are constantly, and I mean constantly, scanning the DNA, proofreading for errors. When they find one, they snip out the bad section and replace it with the right one. It's a non-stop quality control process that keeps the blueprint safe. So, why does all this tiny molecular detail matter so much? Because knowing this code is the bedrock of all modern medicine and biotechnology. It's how we can test for genetic diseases. It's the science that gave us mRNA vaccines. It's the key to gene therapy, creating better crops, and even solving crimes with forensics. It is fundamentally changing our world. And all of this leaves us with one last profound thought. For pretty much all of human history, we've only been able to read the code of life. But now we're learning how to write it. As this technology gets more and more powerful, it raises a massive question for all of us. What responsibility do we have now that we're becoming the editors of life itself?